Proteins are a type of biomolecule, and they have many functions in living organisms. They are structural components of cells. They transport particles into and out of cells. They play a role in the defense against diseases. They make up the major component of skin, hair, and muscles. In addition, proteins are messengers and receptors for cells. They act as enzymes, which help to control chemical reactions in living organisms. And they're made up of building blocks called amino acids. When we look at these proteins are made up of the elements carbon, hydrogen, oxygen, and nitrogen. Some might also have sulfur and phosphorus. So let's look at the general structure of an amino acid. And an amino acid starts with a central carbon. We know that carbon atoms can make 4 covalent bonds. So the central carbon in an amino acid is going to be bonded to a hydrogen. It's going to be bonded to this NH2 group, which is referred to as an amine group. It's going to be bonded to a COOH, which is an acid group. And it's going to be bonded to some kind of a group which varies from amino acid to amino acid. So literally when we look at the structure, it spells out what it is. It has an amino group and it has an acid group bonded to the central carbon. So it's an amino acid. Now when we look at amino acids, there are 20 types of amino acids, and they're categorized lots of different ways. But one way that they can be categorized is by saying that some of them are polar and charged, which means that their R group, which is the part that differentiates every amino acid, is fully charged. They can be polar and uncharged, which means that they might have a partial positive or negative charge, but they don't have a full charge. A third category is non-polar or hydrophobic. So if that R group is a structure that does not have any polarity to it at all, we would classify it as a non-polar amino acid. And the fourth category is when that R group is very small. So let's just look at 3 examples of amino acids. We have leucine, serine, and aspartic acid. And if we look at all 3 of these amino acids, they have that general structure of an amino acid. Each of them has a central carbon atom bonded to a hydrogen, an amine group, and an acid group. The difference between these 3 amino acids is what their R group is. And if we look at these 3 examples, leucine has a non-polar R group. So we would refer to it as a hydrophobic amino acid. Serine and aspartic acid have polar ends to their R group. So they would be referred to as hydrophilic amino acids because they're going to be attracted to water molecules. Now when we look at how amino acids bond together to form proteins, we call the collection of amino acids a peptide. So when amino acids are bonded to one another, we have a peptide. We can have dipeptides when 2 amino acids are bonded together, and we can have polypeptides when many amino acids are bonded together. So let's look at how this process of bonding amino acids together to make proteins occurs. And it should be familiar because it's the same general process that we saw when we took monosaccharides and built them up to make polysaccharides, and when we took fatty acids and glycerol and built them up to make lipids. So we've got an amino acid here, and it's just a general amino acid, so we'll put an R group. We've got a second amino acid here, and we've got this general R group. And what's going to happen is this dehydration synthesis process. So again, we're going to lose an OH off of the acid group of one amino acid, and we're going to lose the hydrogen off of the amine group of the second amino acid, and that hydrogen and OH is going to form a water molecule. So there's our dehydration part. And from there, we're going to need to bond the carbon and the nitrogen together. And that kind of bond between the carbon in one amino acid to the nitrogen of another amino acid is called a peptide bond. When we look at proteins, proteins are made up of four structures. Our first structure is called our primary structure. And the primary structure is the sequence of amino acids that are linked together by that dehydration synthesis process to make a long polypeptide chain. Now, once that primary structure is formed, we're going to have what's called our secondary structure. And what will happen now is this primary chain of amino acids will start to fold on itself and form an alpha helix or folded pleats. And it'll be stabilized by hydrogen bonding between the amino acids. Now, once that secondary structure is formed, we have what's called the tertiary structure. And here what'll happen is once the primary structure is formed and the alpha and pleated sheets are formed, the molecule will start to fold over on itself. And it'll form this tertiary structure, which is the overall three-dimensional shape of the polypeptide. Now, the last structure that we have for a protein is what we call the quaternary structure. And when we look at this, it's basically going to be formed from the association of two or more polypeptide chains. So proteins are made by four structural processes. First, the primary structure, then the secondary structure, then the tertiary structure, and then the quaternary structure.